Now it's going to be important that you take a little time and practice replicating, transcribing, and translating the nucleotides. For replication, we simply have an original string of deoxyribonucleotides, and again, you simply need to pair up or match up the appropriate complementary nucleotides so you have AT or GC pairs. So here we have an example of a string of nucleotides. There's nothing special about them. You can go online, find nucleotide sequences for real organisms, practice using those. Just need to practice, just short sequences. There will be a question of each type of mechanism on the exam that's coming up when we cover and test on genetic engineering and microbial genetics. So here we have our sequence of nucleotides. If you go to the next slide, you'll see how the complementary sequence should look or appear when we replicate this string of DNA. Now, create your own sequence, practice on your own, and you'll find that this is not difficult at all. You just have to remember the AT-GC pairing. Here we can practice transcribing. Again, we're going to start out with our original DNA copy, but now we need to make an RNA copy of the DNA. Again, the original practice sessions or samples in textbooks, online, or on exams won't already automatically be divided up into groups of 3. We're going to need to do that ourselves with our pen or pencil. And in fact, the reading frame for all of our examples are going to be from left to right, just like we read text in a book or a newspaper or a magazine. Now, in truth, it does matter if this is a 3' or a 5' end, and the specific orientation of the DNA is important. But for our examples, we're going to already presume that we have the correct orientation and that we are going to be reading or following the proper sequences from left to right. Realize that if you take a genetics class or you take an advanced course in microbial genetics, your professor will ask you to learn more specific information about the orientation and the proper complementary nucleotides. Right now, we're just concerned about matching up the complementary nucleotides, and we're not going to concern ourselves with 3', 5' complementary or template DNA. So take a look at the example and see if you could also, using a different sequence, transcribe a piece of DNA into RNA. Here's our last example of transcribing and translating. We're actually going to take an RNA copy and, using the genetic code, determine which amino acids are going to be needed for each codon that was created when we created the mRNA transcript. It's simply matching up the proper codon on the genetic code and picking out the correct amino acid from the template, from the original little genetic code. Very easy to do, and you're not going to have to memorize the abbreviations for the amino acids. All of that information will be supplied on the exam, wherein we will have questions and practice sessions of replication, transcription, and translation.